This is the second video in our review of Energy for Life, and in it we're going to do a quick review of some of the things we talked about in video 1, and then we'll look at an overview of the breakdown of carbohydrate from start to finish, and then we're going to look at the two ways that you can provide energy for the resynthesis of ATP anaerobically. And so here we go with that. And so again, humans need energy to do biological work, and there are three types of biological work. The one that's most obvious to us is mechanical work, because again, anytime any muscle contracts, mechanical work is done, energy is required to make that happen. Transport work means that we're moving something against its concentration gradient. That requires a protein in the membrane typically, and it's going to need the input of energy. That's what we refer to as transport work against the gradient. So it's going from low concentration to high concentration. And then lastly, the third type of biological work is chemical work. We're creating covalent bonds. And so anytime we do dehydration synthesis, then we've done chemical work. Perhaps the best example of that would be translation, where we're taking amino acids and building them into polypeptides or proteins. Again, ATP, hydrolysis of ATP, is how we get the energy to drive most, but not all, biological work. And the hydrolysis of ATP is exergonic. That means we have the release of energy, which again is going to be put in to make the work happen. So that's what's going on. And here again, you can see a quick view of the structure of ATP. It is a nucleotide. And so it's a relatively small molecule that is incredibly important in energy metabolism. Just quickly, I'll remind you that we have these two high energy covalent bonds that are holding on the terminal phosphates. And so the truth is that we could in fact hydrolyze both of them. We typically don't do that. We typically just hydrolyze the covalent bond holding on the third phosphate. So usually we go from ATP to ADP. Again, it's important that you understand that cells have a limited stores of ATP. And so you are constantly replacing ATP. You're taking the ADP and you're putting the phosphate back onto it. And if you think about it, you'll realize that that is actually chemical work. We need to put energy in to make that happen. Okay, again, we talked about the first law of thermodynamics. You can't create energy, you can't destroy it, but you can change it from one form to another. The energy that you get in your body is coming from the foods that you eat, the carbohydrate, the lipid, the protein, and so those are all examples of chemical energy. Now, ultimately, most of the energy for life on Earth comes from the sun. And I say most because the truth is that more recently, the past 30 years or so, 40 years, we've learned that there are organisms that live deep on the ocean floor where no sunlight penetrates. They're able to harvest energy from magma that's coming up through thermal vents in the floor of the ocean. So that's why I say most, but not all. And so here again, we reviewed quickly photosynthesis. And so again, photosynthesis is taking energy from the sun and using it to drive the creation of biomolecules from CO2 and water. And so again, the leaves take up CO2 from the atmosphere, and the roots take up O2 from the water. And then we build these complex biomolecules, not just carbohydrates, but also protein and lipid. And then again, a byproduct of that whole process is production of oxygen. Now, we go ahead and we turn it around and we break down carbohydrate in the presence of oxygen, and we produce energy and have CO2 and water as byproducts. Well, now we're looking at cellular respiration. Now, again, we replace ATP as soon as we use it. And the fastest way to use that is to use the substance called phosphocreatine. Well, look at it. Wait, right here again, we have this covalent bond, which is a high energy covalent bond. And so hydrolyzing phosphocreatine, again, is exergonic. And we use some of the energy from that to go ahead and add a phosphate to ADP to give us more ATP. Well, not surprisingly, we're left over with creatine. And so again, as I just mentioned to you, synthesis of ATP, or more accurately, the resynthesis, just simply putting a phosphate onto ADP, is chemical work. Got to put energy into it. Well, here you can see the chemical structure of phosphocreatine. I don't want you to memorize it. It's not critical. But there you can see in the circle is how they're symbolizing that high energy covalent bond that is holding on the phosphate group. Look, it's highly polar, and so it's really pushing away here. Okay. And then, in fact, here you can see kind of the link between the phosphocreatine and the ATP. And so, again, anytime any muscle contracts, we hydrolyze ATP to make that happen. Here you can see that we hydrolyze the phosphocreatine. This is the enzyme creatine kinase, CK, that is making this reaction happen to put the phosphate back on and give us more ATP. Well, you know, later on, the way that we get the phosphocreatine back is, we actually hydrolyze ATP to make that happen. So when we create ATP from oxidative phosphorylation, some of it is used to phosphorylate the creatine to give us phosphocreatine again. Very, very fast. And so, again, you can look at this as one or two reactions. Many people will say it's two reactions, but either way, it's going to be really, really quick because it is only two reactions. All right, here we're looking at an overview of the complete breakdown of glucose. Here it is. And so here we start with what we refer to as stage 1 is glycolysis. Now you need to know a little bit more detail than what is there, and we'll go over that. But you can see a couple of redox reactions happening in glycolysis where you reduce NAD to NADH. Stage 2 is really pyruvate oxidation. I should emphasize again that glycolysis is occurring in the cytoplasm. Now again, stage 2, pyruvate oxidation. And again, as you can see, that's a redox reaction. And here is where we create the acetyl-CoA that we need to go into the Krebs cycle, which, as you see here, is stage 3. And again, we've got a number of redox reactions that are happening in the Krebs cycle. And all of those redox reactions are providing reduced substrate to go on to electron transport. And I hope that you remember that the one and only job of electron transport is to create a proton gradient, which is used to drive oxidative phosphorylation. And so here again, there is one of the byproducts, O2. Most of the CO2 is coming out here from the Krebs cycle. But again, we're looking at cellular respiration, and so byproducts will be CO2 and water. And so again, Krebs cycle, and then these two. We almost always, at least I do, say electron transport and oxidative phosphorylation together because the majority of the time they are indeed linked. Okay? All of the rest of this, pyruvate oxidation on down, occurs in the mitochondria, and much of it is occurring in the mitochondrial matrix, although ETC and oxidative phosphorylation occur across the inner membrane of the mitochondria. But again, as you can see, the oxidation of glucose, that means taking it all the way to CO2 and water, is exergonic. And we're able to capture much of that energy to resynthesize ATP. We don't capture all of it. Much of it is lost as heat. And the majority of that time, that heat is used to help keep your core temperature at about 98.6, roughly. Okay? Again, what we have here is a breakdown of foodstuffs, glucose, lipid, protein, that is exergonic, and so again the cogged wheels are turning clockwise. The creation of ATP from ADP and phosphate is endergonic. It's the smaller cogged wheel turning counterclockwise, and so as this wheel turns, it drives these other wheels. And then the hydrolysis of ATP is used to do work. You could label this as biological work. And then wheel number 1 could be mechanical work. Wheel number 2 could be transport work, and wheel number 3 could be chemical work. And so that works out very nicely from that standpoint. But again, hydrolysis of ATP is exergonic, and transport work is endergonic. Have to put in energy to make it happen. All right, well now here we can see glucose. This is specifically blood glucose. So look, here's the endothelium. And so this is the blood plasma. Glucose is going to move across into the interstitial fluid, and then it's going to use facilitated diffusion and this membrane protein, a GLUT molecule, to move across into the inside of the cell. And again, the thing that happens to it, as soon as glucose gets in there, it gets phosphorylated, becomes glucose 6-phosphate. Much of that will go through glycolysis, but it partly depends on the situation. And so we can go one step further too, and we can convert the pyruvate to lactate. And I don't like the term anaerobic metabolism. Glycolysis is always anaerobic. It doesn't matter whether you end up with pyruvate or lactate. And so if you go to lactate, it's fast glycolysis. But again, that's happening in the cytoplasm. Okay, and so there you go. Now we're going to look a little bit more at the glucose coming in. And so that's what we have over here, blood glucose. It moves into the interstitial fluid, and then it's going to move through the GLUT molecule to get into the inside of the cell. I don't care what cell it is. What we're going to do is we're going to hydrolyze ATP to stick a phosphate onto glucose. Think about it. That's chemical work. We went from glucose to glucose 6-phosphate. Now, if you're at rest, you're playing a video game, you're watching TV, you're sitting there listening to a lecture, much of that glucose is just going to be stored. You don't need it. And so what we do is we take the glucose and we move it from the 6-carbon over to the 1-carbon, and then we store that on the growing glycogen molecule. And so not surprisingly, we're going to say, oh, that's glycogenesis. Glycogen synthesis, all right? Now, on the other hand, if you're exercising, you're going to need even more glucose. So some of this glucose that's going to come in here will continue on down through glycolysis. And in fact, you will even break down the glycogen molecule through glycogenolysis to provide even more glucose. Now, the neat thing is that your liver and a few other tissues, actually, even though it says only liver, actually have an enzyme that will clip the phosphate off so that now the glucose can exit the cell, go through a GLUT molecule and get back out into the blood. And again, you know, one of the jobs of the liver is to try to help maintain blood glucose, keep it from dropping too low. Okay, and so in that case, this would be a hepatocyte. But as you can see over here, oh look, some kidney cells and intestinal cells can do a little bit of that as well. Okay, and so here we have the glucose that becomes glucose 6-phosphate, and we can store it or we can use it for breakdown. And so let me see here. All right, here's the glucose coming in. I know that because here's the hexokinase, hydrolysis of ATP, so now it's glucose 6-phosphate. Move the phosphate over to the 1-carbon, go ahead and add it onto the glycogen. And over here we have an energy harvest. So now we're going to go ahead and get back to glucose and breakdown. So glycogenesis, glycogenolysis. And I just want to remind you that if this is a cell, this must be the phospholipid bilayer. This is a phospholipid bilayer. All right, now we're going to go ahead and look at glycolysis. Here's the glucose. And again, as I said, as soon as it comes into the inside of the cell, bam, we're going to hydrolyze an ATP and we're going to make it into glucose 6-phosphate. And so that's what we have right here. Don't know how well I can write that. You know I'm pretty bad at this, but we'll give it a shot anyway. Okay, well then we're going to use an isomerase and we'll change glucose 6-phosphate into fructose 6-phosphate. So we didn't change the chemical makeup of this thing at all. We just rearranged it a little bit. Well, then as you can see here, we're going to do chemical work again, and we're going to add a second phosphate. Well, got to hydrolyze a second ATP, and that gets us to fructose 1,6, 2 phosphates, bisphosphate. Oh, there you go. And what we've done here is we've bumped up the energy level. So fructose 1,6 bisphosphate has more energy than glucose has. And so for that reason, we refer to this initial part of glycolysis as activation. Well, now here you can see, oh, what do? We take the fructose 1,6 bisphosphate, we chop it in half. And so for that reason, we refer to it as cleavage. Maybe some of you know what a meat cleaver is, and you've seen that. And we chop the meat and do that. Now each piece, so now what we have is we've got a 3-carbon piece with a phosphate on it. We've got a second 3-carbon piece with a phosphate on it. And they both continue on through glycolysis. Oh, look, a redox reaction. We're taking electrons and we're going to reduce NAD to NADH, or send it off to electron transport, which is what we always do with NADH, if we can do that. We go through some other reactions, and now we're finally going to start to get some ATP. Up until now, all we've done is we've lost energy. So now you can see that we're going to go ahead and phosphorylate a DP to get ATP. The interesting thing that you don't see here is there's a compound with a high energy bond right here. There's also a compound with a high energy bond right here. So you can see, oh, look at that. We get 1, 2, 3, 4. But two of those only go back to pay back the ATP that we initially used in the activation stage. So we get a net gain of 2 ATP. And the final product of slow glycolysis is pyruvate. We get 2 pyruvates. Look, up here we've got 6 carbons. 1, 2, 3, 4, 5, 6. Down here we've got 6, but it's in 2 3-carbon pieces. No phosphates. And so because both of the 3-carbon pieces go through the same process here, we refer to this as the common pathway. And because we end up with 2 NADH and 2 pyruvate, as well as our 2 ATP, we say this is slow glycolysis. Now, sometimes you go through fast glycolysis, and we have to add reaction number 11. And in reaction number 11, pyruvate actually goes through a redox reaction in the reverse. We take our NADHs and we go ahead and give those electrons back to pyruvate, and we end up with lactate. Now I want you to notice this is not lactic acid, it's lactate. Not quite the same thing. Okay? And the truth is that, again, we're going to have 2 pyruvates, so we're going to need to use 2 NADHs, and we're going to end up with 2 lactate. And this is what is known as fast glycolysis. The enzyme that does that, you should know it. I make my A&P students know it, is lactate dehydrogenase. It's a dehydrogenase, and so I know it's a redox reaction. So there you go. These are the 3 parts of glycolysis. I want you to know this. Activation, bump up the energy level, and that requires the investment of ATP. Cleavage, chop it in half. Common pathway. This is where we finally start to harvest some of the energy that's in the glucose molecule. But as we'll see in the next video, pyruvate still has energy in it that we can get out by breaking it down further. Here, this is just illustrating for you how we take the energy of the glucose molecule and we bump it up in that activation. Again, we add a phosphate to get to fructose 6-phosphate. We add a second phosphate to get the fructose 1,6-bisphosphate. And then finally, as you can see, greater energy than glucose. And now we're going to break it down to the pyruvate, and we're going to harvest some of the energy. 4 ATP, but we've got to pay back the 2 we used. So the net gain is only 2 ATP. And more specifically, oh look, we ended up with pyruvate, we ended up with 2 NADH. So this is slow glycolysis, slow glycolysis. And then we'll just quickly look at glycolysis. I don't want you to know every single step, but I'm going to just show it to you so you can kind of get an idea what the heck's going on here. And so again, the glucose enters the cell. This is my very poor attempt at showing you a phospholipid bilayer. There's the GLUT molecule. Oh, look, phosphorylated. Phosphorylated again. So we're going to get to fructose 1,6-bisphosphate, bis, not di. And so this is activation. Then we chop it in half. Oh, look, we get 2 pieces that are the same. 3 carbon, 1 phosphate. 3 carbon, 1 phosphate. And so we also went through cleavage. And then finally, we go through the common pathway. And so again, you can't see it, but this compound actually has a high energy bond holding the phosphate group on. And we're going to hydrolyze it. And that's where we will get the energy to do the chemical work of adding phosphate to the ADP. And then phosphoenol pyruvate also has a high energy bond. And so both this compound and this compound have a high energy bond. We hydrolyze those in order to get the energy to resynthesize ATP. We end up with pyruvate. And so again, it is indeed slow glycolysis. Okay? And so I do want you to be kind of familiar with activation, cleavage, and then the important parts of what's going on here in the common pathway. And you should know, final product is pyruvate. Final product is pyruvate. This should have been review for you. In the next video, we're going to go ahead and we're going to finish the breakdown of pyruvate and see what happens there.